Hello, my name is Brooke Chow, and today we are going to be talking about photosynthesis, which is chapter 10 in our biology textbook. Life on Earth is solar powered. What does that mean? Well, chloroplasts capture light energy and convert it into chemical energy, which is stored in sugar and other organic molecules. This process is called photosynthesis. An organism acquires the organic compounds it uses for energy in carbon skeletons by either autotrophic or heterotrophic nutrition. Autotrophs sustain themselves without eating or decomposing other organisms. They make their organic molecules from inorganic raw materials obtained from the environment. They are also known as producers. Heterotrophs live on compounds produced by other organisms. They're also known as consumers or decomposers. So, what are the different parts of the plant and how do they function to photosynthesize? As mentioned, plants have chloroplasts to convert light to chemical energy. They are found in the tissue in the interior of the leaf called the mesophyll. The color of the leaf is because of chlorophyll, which is a green pigment within chloroplasts that absorb light. Then, there are the thylakoids, which can be thought of as flattened sacs inside a chloroplast that are bounded by pigmented membranes on which the light reactions of photosynthesis take place, and these are arranged in stacks or grana. Lastly, there are the stomata, which are microscopic pores that allow CO2 to enter the leaf and oxygen to exit. As you can see here, this is the formula for photosynthesis. However, it's not necessarily this simple. It's actually composed of two processes, the first being light reactions and the second being the Calvin cycle. In light reactions, light is absorbed by chlorophyll, which drives the transfer of electrons and hydrogen from water to an acceptor called NADP+, which temporarily stores energized electrons. Essentially, solar energy is converted to chemical energy in the forms of ATP or NADPH. When ATP is generated with the addition of a phosphate group, it's called phosphorylation. All of this occurs in the thylakoids of the chloroplast. Now, let's talk a little bit about light. A substance that absorbs visible light is called a pigment. Different pigments absorb lights of different wavelengths. A graph that plots a pigment's light absorption versus wavelength is called an absorption spectrum, while an action spectrum profiles the relative performance of the different wavelengths. As shown in the absorption spectra, one type of chlorophyll is chlorophyll A. Other pigments in the thylakoid membrane can absorb light and transfer the energy to blue-green chlorophyll A, which then initiates the light reaction. One such pigment is chlorophyll B, which is yellow-green. Carotenoids are another accessory pigment, and they are hydrocarbons that range from yellow to orange. So, chlorophyll is organized along with proteins and other kinds of smaller organic molecules into photosystems. It can be thought of as an antenna complex. The thylakoid membrane has two types of photosystems, photosystem 1 and photosystem 2. Photosystem 1 is best at absorbing light of wavelength of 700 nanometers, while photosystem 2 has a peak at 680 nanometers. These two pigments are actually identical chlorophyll A molecules. During the light reactions of photosynthesis, there are two possible routes of electron flow, the first being non-cyclic electron flow and the second being cyclic electron flow. As you can see in the diagram, cyclic photophosphorylation produces only ATP, while non-cyclic photophosphorylation produces both ATP and NADPH. Whether photophosphorylation is driven by non-cyclic or cyclic electron flow, the actual mechanism for ATP synthesis is the same. As mentioned, the second part of photosynthesis is the Calvin cycle. Essentially, carbon enters in the form of CO2 and leaves in the form of sugar. The Calvin cycle spends ATP as an energy source and consumes NADPH as a reducing power for adding high energy electrons to make sugar. The carbs produced directly from the Calvin cycle are G3P, also known as glyceraldehyde 3-phosphate, a 3-carbon sugar. There are actually three phases of the Calvin cycle. The first phase is carbon fixation, and the second phase is reduction. Lastly, the third phase is also known as regeneration of the CO2 acceptor of ribulose phosphate. The carbon skeletons of 5 molecules of G3P are arranged by the last steps of the Calvin cycle into 3 molecules of ribulose phosphate. 3 molecules of ATP are spent, and that ribulose phosphate is now prepared to receive CO2 again, and the cycle continues. The G3P spun off from the Calvin cycle becomes the starting material for the metabolic pathways that synthesize components such as glucose and carbs. Now that glucose and carbs have finally been synthesized, that completes our explanation of photosynthesis today. As abstract as everything may sound, photosynthesis is simply a natural process that we wouldn't be able to survive without. I hope you learned something from my video today because I thoroughly enjoyed making it. Lastly, I want to thank Ms. Richards for making AP Biology such an educational and entertaining class this year. Without her help, this video would not have been possible.